Thank you again for bearing with me. Part 2 was definitely focused a bit on the pictures. It's a little unfortunate, so squint as hard as you can, I guess. I don't know.
You guys can come up closer if you want to see them.
Yeah. So part 2, I'm going to be presenting some novel finds for the county. These are mushrooms that I've found over the past year or so, a lot of them from actually just the past couple of months, that were the first observations on iNaturalist of the mushroom in the county. So,
Welcome.
thank you. But the point of this is keep an eye out for them. Look at the small things, slow down, and just notice. Take it slow. First one we have is metarrhizome. This is a fungus that lives naturally in soil, but it also grows on bugs. Yeah.
That's a bug, right?
Yes, this is a beetle. Yeah. So they have a particular affinity for beetle larvae and also beetles. It occurs naturally in soil and is also used in agricultural industries for pest mitigation and control. What's interesting about this fungi is that studies have shown that it interacts with UV light and that UV light can impact its life cycle. Every time I've found this mushroom, which has been once, but other observations of this mushroom when it's growing on bugs have been under rocks and under logs. So this is just kind of speculation, but there might be something going on there with them fruiting on bugs when they're under logs.
These bugs are alive, or is it like a cordyceps?
The bugs are already dead.
So, okay, all right.
Yeah.
Didn't look like a very lively bug.
I post them like that. Dubulia. This is another entomopathogenic fungus. Ento meaning bug, pathogenic meaning pathogen. Does kind of look like a rooster. Yeah, this is a bug that is specific to spiders. It's not currently known to grow on other arachnids. It was described originally in 1897, I believe, 1894. It's found globally, temperate, subtropical, tropical regions. It's often pretty difficult to identify the host that it's growing on. I have a specific statistic from 2024. There were 394 spiders found with just this Dubulia fungus on it. 77% were ID'd to family level, and 23% were unable to be identified. And this is kind of tricky because often the host is what's important for getting this fungi identified down to species. Otherwise, of course, you could use microscopy, DNA analysis. That's kind of an overarching theme, is that oftentimes you need to analyze the actual specimen. And that's important. That's, again, why curation and collecting is important.
So what kind of spiders do they like?
All kinds of spiders. I know that there's one that was actually recently named after David Attenborough. That one grows on orb weavers. But I don't know specifically, but orb weavers is an example of the kind of spider that they do grow on. This is a size reference. This is actually a Morgan. It's tiny, and it's even tinier because it's on my laptop screen. So it's pretty small, and a lot of these fungi that I'm presenting right now are probably more common than we know, but just they're so tiny that they're overlooked. This was actually found by an intern of ours. And on my birthday too, which is pretty cool. This is a species called Sarcoleosha. This is actually an undescribed species. If you see up here species CA01, that's a provisional name. That means it was found in California, and it's the first record of the species in California. It's presumed that this species associates with vaccinium or huckleberry, but it's kind of unknown. A lot of fungi, their ecology is just unknown. We don't really know what they're doing in the environment. It's also maybe speculated that it's growing with moss or biophytes. Yeah, this is an undescribed species. It's related to earth tongues. It's in the Geoglossacea family. Earth tongues are those little black, sometimes green, sometimes yellow fruiting bodies that look like tongues. So they're closely related. This one's pretty small. For example, it's about 2 inches tall. It's another fungi that's probably more common than we know, but just underdocumented due to its small size. And this was first introduced to me by Julian Pimenta, who some of you probably know. This is a size reference. That's the tip of my finger. That's it in my hand. So it's pretty small. That leads to why it's so overlooked.
Wow.
Saccobolus. This is a coprophilous fungi. Coprophilous meaning dung-loving. This one was found on a single piece of deer dung. And if you've seen deer dung before, you know how tiny it is.
Yeah.
So I hope that puts into perspective how absolutely tiny this little mushroom is. I didn't even notice it actually, but I took it home because I found a bigger mushroom growing right next to it. And then I was looking at it under a microscope, and I was like, what's that little thing? And then I did some microscopy, and I was able to identify it as species. And I realized it was the third observation of it in California. But again, it's probably more common than we know, but there aren't a lot of people taking deer dung home. I don't know why.
Looking at it under a microscope.
And then looking at it under a microscope. I don't know why people aren't doing that. Personally, I think we should be doing more of that. But there's actually a lot of biodiversity on dung, and it's a super interesting area of study. These are what the spores look like. Can't really see that well, but they grow in these little spore clusters. A lot of ascomycetes, which is the phylum that this fungus is in, their asci, which is where the spores are kept, tend to be like cylindrical, like elongated cylinders. But what's cool about this specific species is that the spores kind of overlap. So you kind of need like a three-dimensional microscope to look at them almost. Not gonna lie, I can't pronounce this one. Centrium papillatum, I think. Storkbill chytrid. This is one I actually found yesterday, and I actually have a sample of it here, if we can want to pass it around to look at.
Oh my goodness.
It's kind of hard to see in this lighting, but we're looking at the little red globules
Oh
on
my
that.
gosh. I've been reading that all afternoon.
Yeah, so keep an eye out for little red dots on it. It's a fungus that's actually related to chytrids. And chytrids are fungi that grow and infect amphibians. You guys were talking about a foray in the Sierra Nevadas. If you ever see snakes or frogs and their skin kind of looks like it's falling off, it might be infected by this chytrid fungus.
Wow.
This red fungus is not a chytrid, but it's in the same family, or same order. So it's closely related. And what else do I have on here? Yeah, it's closely related to the chytrid fungi, which infect amphibians. It's one of the earliest diverging groups of fungi. It means it's ancient, essentially. It grows on invasive plants like storkbill. It's not known.
Retarded can't talk because people beat me up for looking at their hands.
It's not known to
I've been trying
to
get back to my
kill
family
the
for
plant,
9
which
years
is a bit
now.
unfortunate because it is an invasive
haven't
plant.
complained about it.
But it does grow on them, and it forms these little red globules. This photo kind of reminds me of Christmas, red and green. I don't know. It has a worldwide distribution, and it lives mostly in the plant tissue, but also spends part of its life underground in the soil. And it's parasitic, and there's over 200 species in this genus. So it's a pretty diverse genus. Phylosticta. We have two species here, Phylosticta agrifoliae and Phylosticta vaccinii, growing respectively on Quercus agrifolia, coastal live oak, and Vaccinium ovatum, which is huckleberry in the blueberry family. Actually, this one here, Phylosticta agrifolia, was the first observation of it ever on iNaturalist. I had to contact the curators and say, hey, this species doesn't exist on iNaturalist, can we add it? Because it had been formally described in the 1800s from Berkeley. So we do know that it occurs in Berkeley, that's where it was described, but it hadn't been recorded on iNaturalist before. Phylosticta tend to form these patches on evergreen plants. You can identify them because they have this reddish, brownish, sometimes purplish margin. These little black dots, which you can't really see, those are where the spores are produced. They can cause leaves to shed prematurely, so it's not really a great thing. It's a plant pathogen. And yeah, this one was described from Berkeley. This is the one that's growing on Vaccinium. Again, these are both probably more common than we know, just underdocumented. A lot of people simply overlook things like this, but I hope that part of this lecture, or part of what you guys take away from this lecture, is to not overlook the smaller fungi. Euryciphe, Australiana, the crepe myrtle mildew. This is another plant pathogen. Both the fungus and the plant are invasive, and unlike the chytrid fungus, this one actually does kill the plant. Can stunt leaf growth, can stunt stem growth, and can cause trouble flowering. Can be identified by these little white patches, and it's in the powdery mildew family, which they're called powdery because they kind of just look like powder. Yeah, all right, that's the last one. I have some QR codes here to scan if you want to keep up with me on iNaturalist or on Instagram, and I'll pause now for questions. Thank you for bearing with me. This was kind of not what I was expecting, but still great. Yeah. So thank you guys for sitting out here in the cold. I appreciate it a lot. I'll take questions now. Yeah, go ahead.
So you mentioned huckleberry a couple times.
Yes.
Were you specifically choosing that to kind of look for fungi around that specific kind of host plant?
Yeah, I was. That Sarcoleosha that I mentioned earlier. The Sarcoleosha that I mentioned always fruits with vaccinium. So I was looking just under vaccinium, and I just happened to notice that there were some spots on it. And a friend of mine, Justin, had posted an observation of the Phylosticta before, and so I knew to look for it, and I was just keeping an eye out, and I just happened to notice it.
So were you here in Santa Clara or were you up north?
These were all in Santa Cruz County.
Santa Cruz, so here.
Yes, yeah. Mostly, all of them actually were from upper campus at UCSC.
Did you find any black trumpets while you were down there?
I think so. I only know one black trumpet spot on the campus actually. I'm on the lookout for more spots. So
That's it, huh?
Yeah, those are the red little
They
red
too
ones.
like
I wouldn't notice that.
huckleberries.
yeah, so that's the chytrid fungus that was passing around. Again, like most of these fungi, more common than we probably know, just super overlooked. Yes.
So how'd you get into mushroom photography and identification?
Yeah, I go to school at UCSC, and during my first year, I just kept seeing mushrooms pop out of the ground, and I was like, okay, that's beautiful. I'll take a photo. And then it just started with photography, and then it just absolutely evolved into a passion trying to identify them, collecting them, and then I eventually was an intern at the Norris Center, which is where I currently work. My friend took Christian's class also, and she was like, do you want to go on a mushroom walk with me? And I was like, sure. I don't know anything about mushrooms. And I just got completely sucked into that world.
So,
We know the feeling.
I'm sure, yeah.
question.
Yeah.
So, for all these obscure little mushrooms that aren't in any of the guidebooks, what kind of references do you use what's your approach for identifying something?
That's a good question. A lot of, there's one specific book called Fungi of Temperate Europe, Volume 2.
Little.
Damn, I've got volume 1.
Yeah, Volume 1 has most of the macro fungi, the bigger fungi that tend to form
Basidiomyces.
cap and stipe fruiting bodies. Volume 2 has a lot of these weird mushrooms that are smaller and overlooked. Again, fungi of temperate Europe, all those species are European. So that's helpful for genus level identification, but for species level identification, most of these species are probably undescribed. Only one of them has been sequenced so far, and it came back as an undescribed species. Still waiting on sequences for the other ones. But yeah, fungi of temperate Europe, Volume 2. There's some good resources on the internet. Index Fungarium is a good one. iNaturalist also sometimes will.
Some of these
just-
little indescript ones are really hard.
It's tricky,
It doesn't give you a
yeah.
whole lot of choices here.
Definitely, yeah. Sometimes will give a decent suggestion. For this one, it did give a suggestion, and it was
Wow.
and it was accurate. For a lot of the other ones, iNat was like, I don't know, and I didn't know either. So sometimes I'll also just ask my friends who know a lot about the small stuff. Yeah.
Is that your entire process? Like when you find a mushroom, do you go straight to the books or do you try iNaturalist first?
Usually I will just upload it to iNaturalist and see what it suggests. That's not something that's always accurate, so I'll pretty much immediately after that go to the books. There's a couple good websites, as I mentioned, Index Fungarium as well. Off the top of my head, I can't think of any other good ones, but yeah.
Friends are really helpful.
Yes.
Do you know of any relationships between any fungus and the salamanders up at the university? At Salamander City?
Yeah.
That's what the kids called it that I was with.
That's a good question, and that's also an accurate way to describe UCSC Salamander City. Off the top of my head, no, I don't know. Does anyone else know? Relationships, fungi, salamanders?
It's a moist environment where the salamanders are.
Yeah, oftentimes when I'm flipping logs looking for fungi, I do find salamanders. I saw a hand raised over there.
Do you ever go into the caves at the university to look for fungi? Like
I
the birth canal and all that?
A lot of the caves are closed off. I actually recently today was talking with Morgan about going to explore some of the caves to look for fungus, because there's a lot of really unique biodiversity in those caves. And so it'd be really interesting to explore the fungi that grow in there. So, yeah.
Hey Dan, we've got a local foray.
Yeah, go in the caves looking for coprophilic fungus.
Yeah. Do you have any other questions?
Yeah, I had a couple questions. Are you guys doing any sequencing? Because I'm sending them off to Ohio, my specimens.
Yeah.
So if I could do them more locally, and I don't know if you're associated with fundus at all. Sometimes they are also taking samples. And then the other question was, like, how is the center or the fungarium like developing? It sounds kind of young. Like, are you guys doing like future events or seeking support in any way for expanding things? Or?
We're always seeking support. I'll start with the first question, DNA sequencing. We're in the process of getting all of our specimens sequenced through fundus. Some of our specimens have been sequenced already, about a couple hundred of them, but we have a couple thousand, so we still have a lot of work to do. We're sending them off to Hartsinger. He has a really nice PCR protocol, and so we're in the process of getting those sampled. We're not currently accepting specimens just because we have so much backlog to currently work through. I'm assuming you're sending yours to Stephen Russell at the Ohio,
And Hart.
Yeah, and Hart too. Yeah, both excellent places. I think Stephen does it for free. I think he has a team of volunteers. With Hart, I think it's $2 to $3 per specimen. But yeah, and then yes, we always are seeking support in the fungarium. We do outreach events. We have a couple fungus fairs on campus that we table at. And yeah, what was the other part of your question? Was that it?
Yeah, what developments and ways to get involved or help out.
Yeah, you can check out the Norris Center website as well. That has more information about the fungarium and our herbarium. We have a really cool entomology collection with over 70,000 insects too. So it's a really special place. Yeah.
Sure. Thank you.
All right. Any other questions?
Very good.
Thank you guys.
Hey Reagan, thank you so much. Really appreciate the talk. I want to present with you a small token. This is going to pay for the next 150 DNA